Good morning, good afternoon, good evening to everyone, depending on where you are on Earth when this conference is broadcast. I'm super happy to actually participate for the first time in a Tate conference. And today I would like to talk about marine reptiles, but especially about a series of techniques we've been using and developing in my lab regarding 3D morphology. So, how we can use 3D data to actually better understand the evolution of marine reptiles and especially the ones from the Jurassic and the Cretaceous. So, you know that about 251, 52 million years ago, there was an enormous mass extinction at the Permian Triassic boundary. And in the rocks, the marine rocks after this extinction, you start finding strange fossils, such as this one. And it's not a series of pebbles or shells, it's actually the skull of a reptile, a kind of reptile which is entering the aquatic environment to actually hunt and live most of its time there. So it's this one is actually a series of placodonts. You can see a series of placodonts on the slide here, and you find in the middle Triassic rocks a wide range, a wide series of very strange aquatic reptiles in all forms and shapes. And to me, it's what I like to call somehow the beginning of the strange oceans, because it's the big start, a massive start of what's going to be a true phenomenon, which is the fact that terrestrial animals are going to invade sea and aquatic ecosystems to become the top predators in these environments. So we know that this happened actually several tens of times, where we have many lineages that are going to go from terrestrial environment to the aquatic environment, and this is going to result in strong changes in their morphology, in the shape of their body, of their skulls, or of their fins as well. So even though many of these transitions are concentrated into the Triassic, what I would like to focus on now is the Jurassic and the Cretaceous, and it's really the dominance of marine reptiles that we will be seeing in these times. You can see here what I call some kind of a super tree. It's not fully a super tree, but it's just actually a technique where I will be combining different phylogenetic analysis of many different groups of marine reptiles to have like in one tree. It doesn't matter actually the shape of this tree, or actually all these small characters here. It's just to show you the amount of lineages of marine reptiles that are going to be present that we know at the moment in the Jurassic and the Cretaceous. So it's really something, it's really a key phenomenon in marine ecosystems. For example, I've been using this super tree to derive the number of lineages through time for the Jurassic and the Cretaceous. And even though the fossil record is obviously extremely imperfect, especially for all these large marine predators, we can, depending on how we reconstruct these trees, estimate that for at least some part of the Jurassic and the Cretaceous, the diversity of these animals will actually maybe higher than that of today's marine mammals. So it's really something really important that is expected to have a strong effect on ecosystems. So I'm just putting all these slides to say it's important to study these predators because it's going to give us nice insight onto the evolution of marine ecosystems. It's not a continuous, gentle history. What we see in the evolution of these groups is actually it is filled with strong extinctions, strong radiations, conversion, meaning like some actually new forms are going to resemble other groups that have died, for example, in the past. So it's really something really dynamic, and it's especially the case, as you might see, during the Cretaceous, because in the Jurassic it can be argued that we have some kind of stability within the big groups, right? We have ichthyosaurs here in green, we have plesiosaurs in orange, and then marine crocs in purple. But then this is going to change a lot in the Cretaceous, where we see that the share of the diversity is going to change a lot, where actually ichthyosaurs are going extinct. You also have lots of extinction among plesiosaurians, and then you have the rise of mosasaurs and also sea turtles and sea birds. So Cretaceous is interesting because it's going to concentrate a series of changes in the dominance of many of these groups. So this is somehow the starting point of a big project we've started in my lab, which I've been calling it Seascape, where we are going to analyze how these different groups are going to evolve in terms of their morphology. Are they going to resemble one another? Are they going to be different and occupy other ecological niches? So the plan for today's talk would be this one, where actually we'll be looking at first the 3D data, how to get it and why is it important, and then we'll see how we have applied and used our 3D data to better understand the evolution of marine reptiles, and it will focus on plesiosaurians today. So for the last 5 years or so, I guess, we've been using scanners. We have laser scanners and white light scanner in the lab, and we have digitized many marine tetrapods. I guess we have probably a couple of hundreds at the moment. So you can see here some of the results we can get. So we can get really precise model out of these. And it's just a small sample of the things we do have in the lab we have scanned or using in our analysis. It's not just fancy, right? It's important because having like what I call unbiased morphological data is really crucial. What we have found is like sometimes what is published in papers or in figures actually are erroneous. Sometimes you do have like a strange angle to take the photograph. The scale bars are not correct as well. So just to show you this effect, I've been using a scan we have of a polycotylid from the US, which is called Plesiosaurian. On the left here, I've been using like a human or camera-like distortion you might have, and then where I've been also lying the specimen on its flat left side. On the right, you have exactly the same scan, the same specimen with perfectly orthographic view and perfectly horizontal axis, and you can see major differences in the shape. So if these were two pictures actually in a paper, the measurements we would take out of these pictures actually would be pretty different. And we have found sometimes strong differences in what is actually present in a figure in a paper, and then once we get the 3D scan with the unbiased data. There's also something to be said about measurements we take, because we don't have at the moment, and it's sad actually, no gold standards for some measurements, right? If people are talking about skull length, sometimes they use the midline, sometimes they use the lateral side up to the quadrate, and it will be the same for the mandibles actually, if you take into account the rich articular process or not. So it's important to have these 3D data to better understand morphology and how we can actually then extract information from this morphology. So now we'll move on to the second part of the talk, which will be to analyze the evolution of plesiosaurians and how we can reconstruct what is called macroevolutionary landscapes. So you might know that plesiosaurians, they come in two distinct shapes, right? So we have the short-necked ones, you might see on the left, and the long-necked ones you might see here on the right. It's a bit more interesting and complex actually, because this happened several times, where we have groups that actually had long necks and evolved short necks and the other way around. So today we'll be focusing on the short-necked forms, and we have two main clades that belong to this group, which I call polycotylids, and you might see here that we have lots of very nice fossils in the US, and also the pliosaurids, which you might find on the top of the graph. What's interesting is that both these clades actually they come from long-necked ancestors, meaning that these two groups they have evolved a short-necked morphology with a large skull independently from long-necked ancestors. So we feel it was an ideal case where we can test on how evolution is happening for plesiosaurians, and how we can reconstruct maybe or visualize some of the constraints of their evolution. So what we created is some what I call like a strict protocol, because our goal obviously is to approach the truth. So we've been using our 3D models, but also measurements and pictures we've been taking by ourselves, mostly on all the possible taxa we could actually get nice data from. From all these measurements, and all I want to stress here that all these measurements, they are functionally or dietary informative. So like all these measurements are going to be used because they have a very strong morphofunctional or eco-functional signal. We can build like some kind of a matrix with all these data, and how we can compare each of these forms one by one, or two by two actually. We have what we call a distance matrix. We also have phylogenetic information, is going to be important afterwards. Then from this morphological data, we can use what we call ordination methods, where we are going to reduce somehow the dimensionality of all these traits to try to visualize how these animals are different from one another. This can give you like the rectangle you see, what is called NMDS or PCOH, just what is called a morphospace, which is we try to summarize in 2 dimensions the spread of all these animals. So if some animals are close to one another, it means they're actually similar. And if two animals, 2 points are actually far away from one another, it would mean they are very different. They have a very different shape. Then we've been using these data to try to recreate what we call the macroevolutionary landscape. We'll talk about this afterwards. And obviously all these methods are actually just to visualize, right? It's just a morphospace is to visualize. The landscape is also to visualize, and it needs to be backed by a long series of statistical tests to make sure that we are again approaching the truth. So this is what we can get with these ordination methods. So you have 2 axes, which is the 2 main axes that are going to summarize the similarity or dissimilarity of shapes of all these animals. You have a color, which is actually the clade where they will belong. So you have in red and orange, you have the pliosaurids, and in blue, you have the polycotylids. There are also a series of black lines joining this dot. It's actually the phylogenetic tree, which is uniting all these animals. You can imply it, you can print it, let's say, on top of the morphospace to see actually where evolution is dragging some forms, some taxa in terms of morphology. So when we got these results, what was clear is that there is some kind of a strong phylogenetic component to this, right? Because we have a zone with many orange dots, which is where we find the large pliosaurids that we will find from the Middle Jurassic to the early late Cretaceous. And then you have another zone where you have lots of blue dots, where you have the polycotylids. But what was super interesting is that actually you start to have like 2 different shapes. So these animals with a short neck, they won't actually do it the same way, right? They are not going to resemble one another extremely strongly because we see they are somehow separated in the morphospace. But there's something even more interesting. It's like some member of these groups are going to actually be found in the region, the morphological region of the other. It's especially the case for example, this pliosaurid, which is called a thalassophonean, which is like this domain clade among pliosaurids, which is called Luscan, which I described from Russia, and is found well within the region of otherwise polycotylids. And somehow, in the same way, we have this polycotylate, which is called Plesiosaurian, which is found very close or within the space usually defined by the other pliosaurids. So, with this data and with the analysis we have done on these forms, we can now actually have polycotylates which looks like pliosaurids and pliosaurids which looks like polycotylates. So we made a series on tests of these, and it's pretty clear that actually these are a case of very strong convergence among these forms. And so because we have these separation, but also episode of very strong and very clear convergence, so animals from one group going actually to the morphology of the other, this suggests that we have what we call an adaptive landscape there, or macroevolutionary landscape. So here, Professor Ichthyosaurus is going to explain you what is meant by that. And what we call adaptive or macroevolutionary landscapes is actually some kind of a conceptual surface. So it's really a concept, right, that you might see on the bottom here, where the peaks in this landscape would be the optimal morphologies, and the valleys in between the peaks would be regarded as inefficient morphologies. What it actually means is that you have strong constraints driving evolution. You have strong constraints that are going to force animal towards the peaks because other morphologies not on the peaks are suboptimal. They are not very efficient, and so evolution is believed to be strong enough to force animals towards this peak. So there are different ways you can reconstruct these landscapes. Some people have tried to use single values, like for example body size. What would be the optimal body size for a given clade? Some people have used some kind of performance when you, for example, you measure the speed of a turtle, of swimming turtle, for example, and then you can derive for modern turtles and closely related forms maybe an adaptive landscape for this as well. But here we have a lot of values, we have multivariate values, and we have animals which are long dead. So what we thought of a way to reconstruct the adaptive landscape would be actually to use density. If I'm using the density of points in my morphospace, I can recreate some kind of a landscape. The density of points is somehow used as an empirical way to reconstruct the landscape, because the hypothesis we do here is saying if we have lots of animals evolving this shape, it probably means it's efficient, and so it might be a peak in our adaptive landscape. So I wrote some kind of interactive functions that you can play with it. There's a website actually you can play with the landscape. This is what we get in the end. I think I can do this. Yeah, right. So I have a version here. So this is what we can get actually from analysis, and then you have the landscape. Here will be the peak for the long snouted forms, so like polycotylates, luscan, and these forms. And this is the one to the right would be the peak of the big predators, the very large pliosaurids or polycotylates with a series of adaptations towards a diet of very big, large prey that they would tear and get pieces and chunks out of them, right? So yeah, so I like having these kind of object you can turn around and see. Actually, it's so, again, it's some kind of a concept of evolution, but I like to be able to visualize this. I think it's better than just showing a couple of numbers on a table, right? And then lastly, what we have done with that technique and this project, actually, we've been using this as a tool to explore how evolution of these short-necked plesiosaurians is going to happen over time, and thus also over climate and environmental changes. So on all these graphs here, from the early Jurassic to the late Cretaceous, you always have like the main landscape, which in all these shades of gray. And then for each of these time bin, I will just plot the animals belonging to this time bin. So Pliosaurids, they start off actually way off because they start off with a long neck and a short head. So they start way off this morphology, and then by the middle Jurassic, they start evolve what is called big Pliosaurids. So what's interesting is that by the middle Jurassic, we already have some elements of these landscapes because we have long snouted forms and very large predators as well. Pliosaurids keep exploring these big predatory niches in the late Jurassic, and we think we don't have much long-snouted form there because you have an enormous amount of ichthyosaurs and crocodiliforms in the ecosystems there, which do have a long snout. So we think the niche of a long snout are completely filled during the late Jurassic, and might be a reason why we haven't found yet Pliosaurus with very long snouts in the late Jurassic. There's supposed to be an extinction between the Jurassic and the Cretaceous from reptiles. We've shown a couple of papers over the last years that might not be the case, at least for some groups. And it's probably not the case for Plesiosaurians. They keep evolving new morphologies. And what we see, obviously, these are very broad bin, right? If we want to analyze an extinction, we will focus on something much more detailed, right? But what we do have in the early Cretaceous is, again, evolution of long-snouted pliosaurids. We have the first polycotylids as well. And we have the polycotylids radiation that's going to happen during the late Cretaceous, where they're going to explore lots of new morphologies, mainly in the long-snouted forms, but also we've seen with Plesiopleurodon, also in the large predatory forms as well. So with all this data, we can also derive a curve which are not the diversity, but what we call the disparity, meaning how diverse in terms of morphology these animals are over time. So again, it's quite broad brush, but what we do see somehow of an increase from the late Jurassic to the early Cretaceous, and we have somehow stability or even an increase from the early Cretaceous to the late Cretaceous. So these animals were doing quite well and were still exploring lots of new morphology, at least during the early and middle late Cretaceous. So with this, I'm going to wrap up this talk because it's done. So I wanted to give you an overview of what we can gather with 3D data and why it's also important to gather 3D data. Obviously, it's better to have some very nice measurements and that stuff, but it's also very useful for conservation because once we have nice 3D models which we share with museums, all the museums that want it, it also helps having fewer handling of very fragile specimens. So in terms of conservation, of nice specimens, it's also something that I guess is going to be generalized in the future. And so we have lots of projects with these 3D data, but I wanted to show you one of them, which is actually trying to reconstruct the macroevolutionary landscape of short-necked plesiosaurians. So with this, I would like to thank the FNRS and Bellsport for the funding. The team, actually, the team members of these paper we published last year in Scientific Reports will be Jimmy McLaren, Laura Soul, Rebecca Benyon, Patrick Duncan-Miller, and Roger Benson. And finally, I'm thanking, obviously, all these institutions and people for data. And most especially, I want to thank Jean-Pierre for the invitation and all of you for attending the talk, and I hope it's going to spark interesting discussions. Many thanks. Have a good day, and I hope to see you in person soon. I don't know when it's going to be possible, but yeah, fingers crossed. See you and have a good day.